Hi, my name is Jordan, and I have been studying tomato regeneration and maize transformation through bioengineering. I go to Purdue University, and I'm currently working in the Van Eck lab. So for my tomato regeneration, we have been using a GWAS study that concluded that 3 different genes were able to enhance regeneration or are linked with regeneration of tomato plants. So with that, we used these 3 genes, and we're intending to overexpress them in order to integrate them into low regeneration tomato genotypes. The goal of this is basically to increase regeneration potential of genotypes that maybe are recalcitrant and don't regenerate quite as efficiently. So I was able to design and clone a plasmid and also look at expression levels in the tomato plants to be able to identify and amplify the genes of interest. With that, we ended up finding that gene A was the one that was able to work through all of the steps here, and we were able to overexpress this gene in the tomato plants. So the next goal of this is obviously to introduce it into a low regeneration tomato crop to see the genetic mechanisms of that. With the maize transformation, we are using a Nature publication that tested ensimitol concentrations. The ensimitol is a plant inhibitor, especially in maize, and has been used for higher transformation efficiency. So we studied 2 times concentration of the maize crops, of the ensimitol in the maize crops, and found that they have a drastic phenotypic difference compared to the 1x concentration that the Nature article recently published. With that, because we have the 2x concentration showing higher efficiency in reduction of plant growth, we did a fluorescence imaging study where we had GFP-induced agrobacterium and tried to see how the ensimitol affected transformation efficiency. So with that, we found that there actually were increases in the 2x ensimitol concentration for transformation efficiency. And you can see in some of the genotypes here, we have that expression visualized. So it will be really interesting to see if we can get somatic embryogenesis with this and further the study with that. But yeah.
You did a fabulous job. I'm just wondering, are the findings between tomato and maize transferable to each other, or are they very different in that way?
They're pretty different. However, there are similarities in just the process. This is all an optimization of a production. It's a procedure that we're trying to make more efficient, so the manual labor is less intensive. So while they are different in maybe the strategies, the basics of the procedures can be transferable. So.
All right, great job. Thank you.